Hello everyone, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And in this module, we are discussing about the different aspects related to the cell growth and regulations. So far what we have discussed about the importance of the growth and the significance of the growth in context to the prokaryotic or the eukaryotic cell, and how the growth is being regulated by the having the well developed cellular system, right? And we have discussed these regulations in the prokaryotic system and as well as the eukaryotic system. Subsequent to that, we have also discussed about the different types of growth media, what you can use for the prokaryotic system or the eukaryotic system. And then later on, we have in the previous lecture, we have also discussed about the different stages of the cell growth. So where we have discussed about the mitosis and as well as the meiosis. And we have also discussed about the different types of methods, how you can be able to monitor these events into the experimental system. In today's lecture, we are going to discuss about what will happen when the cell is going to be devoid of the growth or devoid of the media, right? And how it is actually going to respond when there will be an adverse conditions for the proliferations. So now when we talk about the growth, right, a cell is actually going to require to the growth, right? The cell is requiring the growth, and growth is linked to the production of energy, right? Or you actually require a lot of energy for, driving those reactions for producing the energy. And the energy is linked to the nutrition, right? So that's why we have discussed in different types of cell culture media how they are actually going to provide the different types of nutrients, so that you can be able to synthesize the different types of biomolecules, so that you can be able to synthesize the DNA, so that you can be able to synthesize the protein, nucleic acid, and so on, right? Now, nutrients is so there could be the condition where there will be a scarcity of nutrition. So when the cell will enter into a stage where it is actually going to have the starvation, or there will be no nutrition, right? Then it will enter into a stage of starvation, right? Now, during the starvation, it is actually going to start using the reserved food material, right? So when I say reserved food material, these are actually the material which are being produced when there is an excess amount of nutrition. So what happened when you are taking the nutrition? The nutrition is going to be divided into two parts. One part which is going to be utilized for producing the energy, so that you can be able to perform the growth and as well as running the different types of metabolic pathways. On the other hand, if the nutrition is excess to the amount of energy what you are utilizing for the growth and other kinds of biological processes, then this nutrition is going to be get converted into the reserve food material, like for example the fat, right? So it is actually going to be get converted into one of the reserve material which is called fat. It also can be get converted into another food reserves which is called as the glycogen, right? Although we are not discussing about the cellular metabolisms, but the nutrition means carbohydrate, right? Nutrition is mostly the carbohydrate and the fat, which is actually going to be utilized for producing the energy, right? With the help of the different types of catabolic pathways, such as glycolysis, Krebs cycle, and for the carbohydrates, and then for the fat, it is going to be beta oxidations, and subsequent to that, it's going to have electron transport chain, right? So if there will be an excess amount of glucose, then glucose is going to form the glycogen, and if there will be excess amount of fat, then it is actually going to be get converted into fat. Now, once you are going to be enter into a stage of starvation, then the glycogen is going to be get converted into glucose, and then glucose is again going to be utilized for producing the energy. Similarly, the fat, which is stored fat, is going to be get converted into the fatty acid, and the fatty acid is also going to be utilized for the starvation. Now, remember that this food reserve material is limited, right? You are actually going to have a limited quantity of food reserves. So, once the food reserves are going to be depleted, right, or it's not going to be present, then the cell will enter into the acute starvation. Then it is actually going to be enter into the acute starvation, or it is not being able to sustain the required amount of energy required for running the even basic metabolic pathway. For example, you require at least the energy for running your lungs. You are require the energy for running your kidney. You require the energy at least for running your heart, right? Then only you can be able to survive. But if you are not being able to even provide the energy for running these, the life-saving processes, then you will enter into the acute starvation, and after the acute starvation the cell will decide whether I could be able to sustain my life or whether I should go and die, right? So after this it will enter into the death pathway, right? Or it will enter into the death pathway. Now when it enters into the death pathway, it is actually going to have the two different modes of entering into the death pathway. One is which is being called as programmed death pathway, or other one is called as the non-programmed, or it is being called as necrosis. This is being called as programmed. So programmed, within the programmed you can have the two different types of pathway. You can have the apoptosis and you can also have the autophagy. Okay, and apoptosis and autophagy both are programmed. That's why they are not going to damage the cell or damage the body a lot, whereas the necrosis, which is a non-programmed fashion, it is actually going to damage the cells, right, or damage the particular organ. So in today's lecture, we are going to discuss about the death pathway and how the death, if it is following the programmed pathway, how it is actually going to occur for the apoptosis and autophagy, and if it is non-programmed, what are the events are going to happen into the necrosis. So what is mean by the apoptosis? Apoptosis means programmed cell death. Its opposite or the antonym is called as necrosis. So whatever we are actually happening under the control of the organism, that is apoptosis. And the programmed cell death can be of two types. It could be the one which is apoptosis, the other one is called as the autophagy. Apoptosis is happening within the cytoplasm, which is actually where you are actually going to start eating your own organelles, and the formation of large vacuole that eat away the organelles before the nucleus is destroyed, right? So remember that in the cell you have the multiple copies of mitochondria, multiple copies of the lysosomes, multiple copies of the Golgi and so on, right? So you have probably the 500 copies of mitochondria. So if you can take or eat some of those copies, probably you can actually be able to recycle the material and sustain for a certain amount of time, especially when you are under the acute starvation conditions. And for the apoptosis, it is actually going to have the set of the reactions which are actually going to drive the programmed death of that particular cell. So apoptosis is a Greek word which is called as falling off or the dropping off. It is a self-destruction programmed genetically sequenced in the biomedical events. It is a kind of a counterbalance by the elimination of similar number of cells, and it is basically being called as death by the design. So when you talk about the apoptosis, you can also be able to talk about the differences between the apoptosis and necrosis, because then only you will understand why the apoptosis is beneficial for the organism and why the necrosis is not beneficial. So when you talk about the differences, the apoptosis is a programmed cell death. So it's an energy dependent programmed cell death, whereas in the case of necrosis, it is going to be where the cell dies due to the damage, right? So if the any cell undergo through the different types of injuries, and if it injury is beyond a limit where the repairing is not possible, then it will enter into the necrosis. Apoptosis may occur into the one cell, the necrosis may occur into a group of cells. The apoptosis does not lead to the inflammation, whereas the necrosis is very important, and it is actually going to be responsible for production of the inflammatory reactions, and that's why it is going to damage more tissue, because when you are going to have the inflammatory reactions, that all I think we are going to discuss when we discuss about the molecular immunology in the later part of this particular course. You will see that the inflammation is going to produce large amount of ROS, large amount of free radicals, large amount of nitrogen centered radicals, oxygen centered radicals, and so on, and that is actually going to damage the neighboring tissue. So it is actually going to damage the large amount of tissue than what is being injured actually. Then there is a central role of mitochondria in in regulating the programmed cell death, whereas there is no role of mitochondria in the case of necrosis. Then in the case of apoptosis, the dead cell are going to be eaten by the macrophages, so it is actually going to be cleared off very systematically, whereas in the case of the necrosis, the dead cells are going to be removed by the macrophages. Apoptosis, where the cell is going to be go through with the process and it's going to be shrinked, whereas in the case of necrosis, the cell is going to swell, which means the cell is actually going to have more amount of inflammatory molecules, and that's why the necrosis is actually called as unhealthy mode of death, whereas the apoptosis is actually a programmed cell death. So in the apoptosis, what will happen is that you have a normal cell, right? And then this normal cell, when it actually going to start catalyzing the events of the apoptosis, it's going to enter into a cell shrinkage, and there will be a chromatin condensations. After that, the membrane is going to be distorted, and then there will be a membrane blubbing, which means it's going to form the blub-like structure. And then after this, you are actually going to have the nuclear content, which is going to be collapsed, and then ultimately it is going to form the apoptotic bodies, and then these apoptotic bodies are actually going to be eaten up by the macrophages. There are many players which are going to participate into the apoptosis. So you can have the apoptosis initiating factors, you're going to have different types of caspases, you're going to have the BCL proteins, you're going to have apoptosome, and then you're also going to have the p53 gene and as well as the p53 proteins. Now talking about the caspases, these are the cysteine proteases, right? And these are specific proteases which are being catalyzing a cascade of reactions to drive the apoptosis. It is a family of cysteine proteases. It is present in the cell under the inactive forms. So they are actually going to be present as the inactive forms. And the inactive forms are actually going to be cleaved at one end, and that's why it is actually going to be converted into the active form. So you're going to have the, you're going to have like this, right? So this is inactive form, and when there will be a signal, right, that signal is actually going to cleave off this particular bond, and that's how you are going to have this, right? And this is going to be the active form, and this active form is going to be a cysteine protease, and the job of this active form would be that it is actually going to activate the another caspases, and that's why you are actually going to have the two classes of caspases. One is called as the initiator caspases, which are actually going to initiate the reactions, and the second one is called as the effector caspases, which are actually going to affect the cells, right? So in the initiator caspase, you are going to have caspase 2, 8, 9, and 10, whereas in the effector caspase, you are going to have caspase 3, 6, and 7, right? And initiator caspase is going to take up the signal, like the death signal, and then they are actually going to catalyze the different types of reactions. whereas the effector caspases are actually going to directly affect a biomolecule. For example, in the case of caspase 3, it is actually going to start degrading the different types of proteins and different types of DNA actually. So it is actually going to activate the DNases, and then the DNases are going to start cleaving the DNA part. So these are the classes of different types of caspases, and this is the structure of the caspases. So you are going to see that majority of these caspases are actually participating into the apoptosis. Some of these caspases are also involved into the inflammatory pathway and so on. So caspases are actually having the two different types of domain. One is called as the caspase recruitment domain, or the CARDS, and the other one is called as the Det effector domain. So these are called Dets. Now this is what I already explained. You are going to have the active caspases, and when the active caspase is going to be formed, it is actually going to initiate the cleavage reactions for the other caspases, and that's how it is actually going to catalyze the cascade of reactions. Then the second molecule is called as the BCL2 family. So it is the regulatory proteins of the mitochondria, and it is the first time gene which is isolated from the B cell lymphoma, which is a cancer, and that's why these proteins are being called as the BCL2. They all have the BH3 domain, like the BLC2 domain, right? And BH3T is a pro-apoptotic domain, and it gets exposed and it gets activated. Now, for talking about the BCL2 family, you're going to have BH124, BH123, BH3, and within the BH14, you are going to have the anti-apoptotic protein, which is called as BCL2 and BCLXL, whereas BH13, you're going to have the pro-apoptotic protein, which are called BAX, BOX, and BAC. And then you have BH3, which is called to be pro-apoptotic protein, that is BAD, BIC, and BID. And what is the function of these BCL2 family? It is regulating the balance between the pro-apoptotic and anti-apoptotic situations. It is involved into the intrinsic pathway, and it is going to induction of the BCL2 in by a Det signal. So BCL2, which is called as B cell lymphoma, BH, which is called as BCL2 homologs, BID, which is called as BCL2 interacting domain, BCLXL, which is called as X or the close homolog, and the L is called as, which is going to inhibit the apoptosis. So BCL2 and BCL2XL, right? Then we have the apoptosome. So it is a multi-unit protease activation complex. It forms after the induction of cell death into the mitochondrial pathway, and the discovery of apoptosome is being done by the Professor Wang, and it is a wheel-like structure connected to the 7 radial spikes. Now, talking about the machinery, now we are going to talk about the pathways which are important for the apoptosis. So you have the two different types of pathway. One is called as the death receptor pathway, which is called as the extrinsic pathway, and then you also have the mitochondrial pathway, which is called as the intrinsic pathway. So intrinsic pathway, the inducers are from the cell. Extrinsic pathway, the inducers from outside. So this is actually the place where the distinction is. So you don't have to remember which reaction is going to induce the extrinsic pathway or the intrinsic pathway. I mean, majority of the cases, the inside, if the cell is not performing well, it is actually going to induce the intrinsic pathway. If it is getting any kind of threat from outside, then it is mostly been going to initiate the extrinsic pathway. For example, if you are treating a cell with drug, right, then that it is actually going to activate the extrinsic pathway. But if you are treating with the drug and the drug is going inside and it is actually changing the microenvironment, then it is actually going to induce the intrinsic pathway. Now let's first talk about the DRESS receptor pathway. So induction is mediated through the cell surface receptor known to be DRESS receptors. So basically on the cell you are actually having the receptors, and some of these receptors are going to respond to the drug molecule, and that's how it is actually going to initiate the cascade of reactions which is related to the DRESS receptor pathway. Det ligands are activating these receptors, and the further activation of caspases which are going to be lead to the cell death. So whatever the machinery we have discussed for the apoptosis is going to be downstream to these initiator pathways, right? whether it is a death receptor pathway or the mitochondrial pathway, the initial the induction is going to be different. The inside the cellular machinery is going to be the same. It is going to use the caspases, it is going to be use the BCL2 proteins, it is going to use the apoptosome and so on, right? Now, within the DES receptors, you are actually going to have the different types of DES receptors. So, this one of such receptor is TNFR superfamily, where you have having the DES receptors like TNFR1, CD95, TRAIL R1, and TRAIL R2, and these are their corresponding the ligands. So, which is one is called as TNF alpha, the other one is called as CD95L or fast ligand. Then you also have the TRAIL R1, and then you also have the TRAIL R2, right? So these are the full form of the TNFR and all that, right? And what happens ideally is that the ligand is actually going to bind the receptor, right? So once there will be an induction of that receptor pathway, the ligand, which is TNF alpha in this case, will go and bind to the that receptor which is present onto the plasma membrane, right? So you see the plasma membrane, right? And so binding of the ligand to the DES receptor is going to initiate the downstream signaling, and this downstream signaling is then actually going to activate the apoptotic pathway, and that's how these are actually going to be extrinsic pathway. So you have the different types of receptors. You have the fast ligands, fast receptors, you have the DR4, you are going to have DR5, you have the TNFR1, you have the DR3 and all that. So these are actually going to be the different types of receptors, and they are actually going to participate into the different types of reactions. Once the receptor is going to bind their ligand, then it is actually going to bind the adapter proteins, the different types of adapter proteins. For example, most of these are going to bind the FADD, and then these adapter proteins are actually going to drive the downstream signaling, and ultimately they are actually going to collect the cellular machinery for apoptosis. So they are going to activate the caspase 8, caspase 10 and so on, and then ultimately it is going to form the apoptosome, and that's how it is actually going to damage the cell, all the proteins, DNA and all that, and ultimately these cells will going to form the apoptosomes, and these apoptosomes are actually going to take up by the macrophages. So this is what exactly it is showing here. In the extrinsic pathway, you are actually going to bind the ligand, and then the ligand is actually going to take up the adapter proteins, and then these adapter proteins are going to activate the inactive caspases like the procaspase 8 and 10 into the active caspase 8 and 10, and then these active caspases are going to activate the downstream effector caspases like the caspase 3, 6 and 7, and then ultimately it is going to activate the production of the apoptosome, and that's how it is going to induce the apoptosis. Now coming to the mitochondrial pathway, so it is going to be release of the protein from the mitochondrial immunal membrane into the cytosol. So inducer is that the proteins from the mitochondria is going to be released into the cytosol, right? And then it is going to induce the DNA damage, and then it's actually the what are the inducer for the mitochondrial pathway? So you're going to have a cellular stress, heat shocks, oxidative stress, cellular damage are actually the inducer for the apoptosis which is following the mitochondrial pathway. So what happened exactly is that you are going to have the cell damage, right? And that cell damage is going to activate the p53, and that is actually going to activate the pro-apoptotic BCL2, and then pro-apoptotic BCL2 is going to induce the release of the mitochondrial factor into the cytosol, such as the cytochrome C. Once the cytochrome C is going to be released, it is going to make a complex with the alpha-1, and that's how it is actually going to activate the caspase 9. So procaspase 9 is going to be activated and form the caspase 9, and that's all these are going to be come together and form the apoptosome, and these apoptosome are then going to initiate a cascade of caspase activations, and that's how it is going to activate the caspase 3, and the caspase 3 activation is ultimately going to lead to the damage of the cellular machinery. So this is exactly what is going to happen. The apoptosome is going to be formed. Then it is going to activate the caspase 9. The caspase 9 is going to activate the caspase 3. And then once the caspase 3 is going to be activated, the caspase 3 is going to induce the apoptosis. So this is what exactly going to happen. You have the healthy cells. It is going to get an injury, right? If the injury is moderate, right? If it is repairable, then the cell will enter into this phase and it will go and get the healthy cells. If the injury is beyond the repair, right, then it is, or it is causing the different types of genetic disorders, right, then it will not be it will not reverse, right, it will not go back, right. And then it could also possible that you got injury first time, then you again got another additional injury, and that's how, if the cell will decide that it will not be repairable, right, then it will going to initiate the caspase activation, and it is going to initiate the programmed cell death, and that's how the cell is going to enter into the apoptosis. So a damaged cell may undergo the apoptosis if it is unable to repair the genetic disorders, right, or genetic errors. Now, what is the significance of the apoptosis? So significance is that it is actually going to delete the dangerous and the damaged cells. It is required for the development of the organs. It is without apoptosis, the human can grow. Whole epithelial cell lining changes every 23 days, right? So basically, the apoptosis is actually regulating the cell growth. So it is not allowing the cell growth to go beyond a limit, right? And that's how it is keep killing the cells. Then it is also required for the regulation of the immune cells such as T cells, and it is also required for the development of the cancer and the neurodegenerative diseases. Although we are not discussing in detail about all this, how the apoptosis is relating to the development of cancer or neurodegenerative diseases, but that we can discuss later on into some other courses. Now, talking about the pathological implications, so growth of the tissue and organ in the embryo, then replacement of the time expired cells like the gut epithelium, then repair and healing after the injury or the inflammations, then regeneration of the tissue and the hyperplasia. So these are some of the events where the apoptosis is taking part actively, where if the apoptosis is not there, then it is actually going to cause the more pathological impact. Then you also have the hepatic implications. So in the neurodegenerative diseases like Alzheimer's disease, the apoptosis is being induced into the neurons, and that's how the neuron, there will be a neural damage. And when there is a neural damage, then it will actually going to reduce the number of neurons, and that's how it is actually going to be responsible for the development of diseases like the Alzheimer's. Then we also have the myocardial infarctions. So myocardial infarction happens when there will be a development of hypoxia. And because of the hypoxia, the some cells are probably not getting the enough amount of oxygen, and that is going to induce the production of ROS. And because of the ROS production, the some cells are going to be damaged into the heart, and that's how it is actually going to cause the myocardial infarction. Then we also have the some of the apoptotic cells or apoptotic proteins as a target for the anti-cancer drug development, because if you can be able to induce the apoptosis into the cancer cells, and how you can be able to induce the apoptosis, because apoptosis is being controlled by two different types of protein. One is the pro protein, the other is the anti protein, right? So you have the pro-apoptotic proteins or you have the anti-apoptotic proteins. Now, pro-apoptotic proteins are going to increase the apoptosis. Anti-apoptotic proteins are going to decrease the apoptosis. So if you increase the apoptosis, you are actually going to enter, or you can actually be able to damage the cells. If you have the anti-apoptotic, then it is actually going to be responsible for development of cancer, because these cells are going to survive. And since these cells are already being damaged genetically, there is a highly likely chances that these cells may enter into the cancer. So if you increase the anti-apoptotic factors, such like some of the BCL2 proteins, then it is actually going to allow the cancer cells to survive, right? and because of that it is not good for the, so some of these cells probably can be used for as the anti-cancer drugs. Then we also have the neurodegenerative diseases. So you can have the Alzheimer disease, which shows the excessive apoptosis. Then you have neural growth factors and brain derived from the neurotropic factors. Then you secrete the protein that maintain the balance between the pro-apoptotic and the anti-apoptotic factors. And then the blockage of these factors are new area of interest for the target drug delivery. Then we also have the myocardial infarctions. So it occurs due to the blockage of the coronary artery by the thrombus, right? And the decrease in oxygen supply by the necrosis or the apoptosis. So you can actually have the cell death by the two pathway, which is called as necrosis or the apoptosis. Then you can also use the apoptosis for targeting the cancer cells. So you can actually use the BCL2 proteins as a target for the new drug development. BCL2 is the anti-apoptotic and increase resistance to the cancer chemotherapy. And the DES receptors and the respective guidelines can be used in targeting the anti-cancer drugs. Now, how you are going to detect the apoptosis into a particular type of cell? So you actually have the multiple way in which you can be able to detect the apoptosis into cells. So you can actually be able to use the detection of the programmed cell death either by the morphology. You can use the molecular assays such as DNA fragmentations, or you can use the flow cytometry and enzyme assays. So detection on the morphology, you know that the classical morphology of the apoptosis is that there will be an induction of the cellular shrinkage. Then you also have the detachment from its neighbor. Like for example, you know the cells are growing like this, right? They are stick to each other. But you can have the cells like this. If you have a cells like this, that means it is actually detaching from its matrix. And that is the clear indication that probably something happening into this. Then you can also have the fragmentation of the nucleus. You will see the nucleus is like this, right? And ideally, you will see a nucleus like this. So this is healthy nucleus. When you see the nucleus like this, that means it is actually inducing the apoptosis. And then you also can have the packaging of the cell into the multiple plasma membrane, which means you are actually going to have a cell like this. You're going to have like this. That is the apoptotic cells. And then you are going to have the phagocytosis of these fragments. So these cells are going to be fragmented. Then you can also have the detection of using the DNA fragmentation. So remember that when I think in the subsequent lecture we are going to discuss about the genome packaging, right? So when you talk about the genome packaging, right? So genome is actually forming a nucleosome, right? So you remember that you are actually going to have different types of nucleosome, and the DNA is unwinding on this, and then it's going like this, right? Like this, right? So DNA is going like this, right? And here you have the H1, right? Which is a linker histone. So when you are exposing this genome to the enzyme, right? So what enzymes are doing is they are cutting this. They are cutting from here, right? They are cutting in between, right? And because of that, the length what is being covered onto or the wrapped onto the nucleosome is going to be released, right? And this is actually going to be come into as the fragments, right? So these fragments would be like X. Then if once you get from here, you're going to get 2X. Then you get from here, it's going to be 3X, right? And because of that, it is actually going to give you a pattern which is called as DNA ladder. So this is being called as DNA ladder, right? Although we don't have enough time for discussing about the process and all those kind of things. So, but when you are going to isolate the genomic DNA from a damaged cell, and when you resolve it onto the agarose, you are going to get a DNA ladder like this, because all these fragments are separated from one another by the length of this particular DNA, right? So and because of that, it's going to form a ladder, right? So you're going to have X, 2X, 3X, 4X, 5X, like that, right? So basically it is going to keep increasing, right? So remember that this length is 180 base pair, right? So it's going to be 180. For this one, it is going to be 360. For this one, it is going to be like that, right? So when agarose gel is going to be used for separating this genomic DNA, you are going to get a DNA fragments and DNA ladders. And this particular assay is called as DNA fragmentation assay. And the applicable of the total cellular DNA to an agarose, the fragments are separated throughout the agarose gel and the fragment will be detected by the ETBR staining. Then you can also use the flow cytometry. Remember that the apoptosis is going to have a lot of changes into the cell, right? It is actually going to change the nucleus, it is going to change the outer plasma membrane and so on, right? And that all we have not discussed, right? So flow cytometry can be used for, to detect these modified cells. So what you can do is you can use the flow cytometry, you can stain the cells with the different types of dyes, and then you can analyze them into the flow cytometry. And we have prepared a small demo clips where the students are actually going to explain you how you can be able to stain the cells with the different types of dyes, which are going to stain the nucleus and as well as the plasma membrane. And that's how you can be able to detect the apoptosis.
Hello everyone. In this video we will be discussing about how to perform live dead cell staining using actinium orange and rubidium iodide on FACS equipment. So the basic principle is that the actinium orange is permeable to both the live and the dead cells, whereas the rubidium iodide is only permeable to late apoptotic and necrotic cells. So this property of actinium orange and rubidium iodide lets us to recognize what population of cells are in late apoptotic or early apoptotic or necrotic cells. So coming to the procedure, the first thing we do is that we trypsinize the cells from the 100 mm cell culture dish and then plate 1 million each in the untreated and the treated well. So after 12 to 14 hours of adherence, we treat the samples according to our requirement. And then let's say that we are treating for 24 to 48 hours. Then after the appropriate time, we trypsinize the cells, collect the pellet, wash it 2 times with PBS, and then resuspend the pellet in 2% fetal proven serum in phosphate buffer saline. So after we have resuspended the pellet in the 2% FPS in phosphate buffer saline, we give the appropriate actinium orange and rubidium iodide treatment. The working concentration for actinium orange is 0.5 to 1 microgram per ml, whereas for the working concentration for rubidium iodide is 1 to 5 microgram per ml. So we add the dyes just before taking the data, or we can just give 10 to 15 minutes of incubation for the dyes to bind to the cells. So that, and after that we acquire the data on the FACS equipment.
So after adding the actinium orange and rubidium iodide to the cells, we have to acquire the data on the CellQuest Pro software. So the first thing we do is open the CellQuest Pro and connect it to cytometer. And then we need the counters, the detector and amps, and the status. For the actinium orange rubidium iodide staining, we need 2 dot plots. One is for the FSC, SSC for the forward scattering and the side scattering, and the other one is for the FL1 and FL3. So the FL1 plot is on the x-axis, whereas the FL3 plot is on the y-axis. The FL1 plot is for the actinium orange and FL3 plot is for the rubidium iodide. After taking the plots, we have to set the directory and save the data in our required location. In the detector and amps, we have to remember that we have to set the population of the healthy cells in the first quadrant, that is 10 to the power of 1 and 10 to the power of 1. After we set the untreated cells in the first quadrant, and then we analyze the treated cells, and then we can say whether there is any shift in the fluorescence in the untreated and the treated cells. For the treated cells in the third and the fourth quadrant, that represents the apoptotic and the necrotic cells. So now we will be taking the sample, but before analyzing the data, we have to set the number of events to the 5000 and then keep it on setup, and first we will see whether the events are coming properly or not. Now we press acquire. As we can see that in the FSC plot and SSC, in the FSC and SSC plot, we can see the events coming near 0, that represents the healthy population, as well as in the FL1 and FL3 we have set the healthy population between the 10 to the power of 1 and 10 to the power of 1. So this represents the first quadrant. We will show in detail how to do the quadrant analysis in the FCX Express 5 software. Now that we have set the population in the first quadrant, we will remove it from setup and acquire the data. After the untreated samples, we have to take the treated samples on the same parameter description which we have set for the untreated cells. Now we change the sample in the sample injection port to the treated sample. We have to remember that we don't have to change the parameters, or else we will never be able to say whether there is any shift in the untreated or the treated cells if we change the parameters. After changing the sample, now we have to choose the directory for the treated cells and then change the name also to treated, and also the file count to 1. Then press OK, and then now we acquire the data on the same parameters. As we can see that there is some shift in the population of the cells. The population is having a little bit more fluorescence than the untreated cells, which represents the apoptotic and necrotic cells in the third and the fourth quadrant. In the fourth quadrant, mostly the necrotic and the dead cells are present, whereas in the third quadrant the late apoptotic cells are present. After we take the untreated and the treated samples, after we acquire the data for the untreated and the treated samples, we have to analyze the data in the FCS5 Express Pro software using quadrant analysis. In the quadrant analysis, we can see how much population of cells are present in which quadrant, and therefore we can identify the number of healthy populations and the apoptotic and the necrotic cells. So after acquiring the data in the FACS equipment, we have to now analyze the data in the FCS5 Express software. So the first thing we do is we open the new layout and change the orientation to landscape, and then now we input the data. First thing we do is we take the untreated file and then open the dot plot. We need two dot plots. The one is the FSC SSC and the other one is the FL1 FL3. The FL1 FL3 dot plot shows the live and dead cell staining. The FL1 is responsible for the acridine orange, whereas FL3 is responsible for the propydium iodide. And now we take the treated file and again we select the dot plot. In dot plot as well, we need the FSC and the FL1 FL3 plot. As we can see that there is a difference between the untreated and the treated sample. Now we have to find out how much percentage of the cells have gone actually the apoptotic or necrotic. Like we have to divide the population of cells into 4 quadrants using the quadrants option. So we go to the gating and then take the quadrant and then apply it on the FL1 FL3 plot. We have to apply it in such a way that we cover all the cells in the untreated plot. So let's say that in the untreated plot we are having 92% live cells and 7% are in the early apoptotic phase. And then after applying the quadrant to the untreated, we have to apply the same quadrant to the treated one in order to find out the difference between the two. So just we click on the quadrant and then we copy and paste on the treated one. So in this way we can say that in the first quadrant in the untreated sample we have approximately 93% of live cells, whereas in the treated one we have, like in the treated one we have only 35.7% healthy, whereas the 33% have gone are in the late apoptotic and 29% are necrotic cells. So in this way we can use acridine orange and propydium iodide to determine the healthy, the apoptotic and the necrotic cells in different treatments. And also we can establish a relation between different concentrations of treatment as well and the number of live and dead cells in any experiment. So this is the way we analyze and process the data on the FACS equipment in order to do the live and dead cell staining.
So I hope you could have been understand the different processes or different events what is required for studying the apoptosis using the flow cytometry, and it may be useful for you to understand the process, and as well as you can be able to use this process in your laboratory. Now let's talk about the another programmed cell death, which is called as the autophagy, right? Or autophagy, where the cell is going to start eating its own organelle. Now autophagy is literally means self, right? Auto means self and phagy means eating, right? So when you start eating your own self, then it is called as autophagy, a process by which the cell break down and destroy the old, damaged or abnormal protein and other substance in its cytoplasm. The breakdown products are then recycled for the important functions, especially during the period of stress or the starvation. So mostly it happens during the starvations. When you remove the essential amino acids, when you remove the large quantity of glucose and so on, then the cell is actually requiring those essential amino acids. So what it is going to do is it is going to start taking the proteins from the cytoplasm, or it is start taking the different organelles such as mitochondria, Golgi bodies, and lysosomes and so on, and then it's going to take up. So these are some of the inducers for the autophagy. One is nutrient starvations. The second one is the response to the hypoxia. The third one is called as ER stress. The fourth is called as the cell death. The fifth is called as cell damage, and the sixth is called as the oxidative. Although these inducers are different, but the downstream molecular events are going to be same, right? Majority of these molecules are, or majority of these inducers are actually going to initiate a particular cascade of reactions, and because of that, it is actually induce the autophagy. Now, when you induce the autophagy is going to follow the different pathways. So you can have the microautophagy, you can have the chaperone mediated autophagy, and you can also have the macroautophagies. In the microautophagy, some proteins and the organelles are directly going to join to the lysosomal vacuole or the late endosomes. Generally, it is a non-selective process, but in that some cases it is selective. For example, the microexophagy and pyxiamental macrophagy of the nucleus and the micromitopathy. So in some of these cases, the organelles are directly going to join the lysosome. Remember that the lysosome is going to have the different types of proteases, right? And also going to have the nucleases. So it is actually going to be utilized for digesting the organelle and the organellar content, and that's how these raw material is then again going to be distributed to the cell for its usage. Then we have the chaperone mediated autophagy. So in some special cytosolic protein have a special guide motif which is called as KERQ with HSC70 and other chaperone attached to the lysosomal membrane. That guide motif attach them to the LAMP-AZ receptor at the surface of the lysosome and transport them into the lysosome with the LAMP-A2 dependent transporters. So in the chaperone mediated autophagy, chaperone is a cellular degradation machinery which is actually going to be utilized for degrading the unfolded or misfolded proteins. So it's basically the protein which are actually going to have this particular motif will actually be delivered to the chaperone, and then this chaperone is actually going to deliver all these to the lysosomes for its recycling. Then we also have the macrophagy. So macrophagy is where you are going to have the nucleation and the extensions. There will be a closure. Then you are going to have the fusion with the lysosomes, and then the lysosome is going to digest the material, and that's how you are actually going to have the recycling of this material. What are the regulators? So there are many proteins which are actually going to regulate the autophagy. such as BCL2, ROS, calcium, AMP activated protein kinases, BNIP3, DRAM, calpain, FADD, and IP3. And we are not going to discuss about their regulatory mechanism and so on, because then it is going to be beyond the scope of this particular lecture, right? One protein what we are going to discuss is called as the mTOR, right? And mTOR is actually regulating the induction of apoptosis in cooperation with the other two nutrient sensing pathway, that is protein kinase A and SH9. In mammalian cells, the MTR appear to regulate autophagy in a similar way to the yeast. The mammalian target of riboflavin or MTR act as a negative regulator, and the extent of autophagy is regulated by the protein upstream of MTR signaling, including the P10, PDK1, AKT, and TSC1 by 2. P10 and TSC1 by 2 positively regulate autophagy, whereas the AKT actually inhibits, right? So it is actually going to induce the apoptosis, whereas this is going to be inhibit. So downstreams of MTR, including the elongation factor 2 and the SX kinase, which is shown to regulate the autophagy. Then we also have the P53. So P53 is a suppressor, is mutated in approximately 50% of the human cancers and induce the autophagy. P53 can be positively or negative regulator of autophagy depending upon its subcellular localization and the type of stress. It has been reported that the P53 function as a critical mediator for the damage-induced apoptosis has been shown to induce autophagy in the damage-regulated, autophagy-regulated dependent manner to execute a full cell death in human cancer cells. DRAM is a lysosomal integral protein and is a direct target of a P53 induced macrophagy and its accumulation of the autophagosomes. Disease related to the autophagy. So you can have the degenerative diseases, you can have the metabolic syndromes, you can have the aging, you have the infectious diseases, then you have the self-immune problems, diabetes, cardiomyopathy, and as well as the cancers. Now, talking about what we have discussed in this particular lecture, so we have discussed about the conditions under which the cell is actually going to be under extreme starvation conditions. And once it is under the extreme starvation conditions, it has two choices. One, that it can go to the programmed cell death, or the second is it can go to the non-programmed cell death or the necrosis. Within the programmed cell death, we have the two different modes. One is called the apoptosis, the other one is called as the autophagy. Apoptosis is going to induce the cell death, whereas the autophagy is actually going to utilize the cellular machinery again and again so that the cell can actually be able to overcome the acute starvations. Majority of the cases, the autophagy is going to be induced first, and when the even autophagy cannot sustain the cells for survival, then only the apoptosis is going to be induced, and that's how the apoptosis is going to kill the cells. And apoptosis is going to be utilized the two different types of pathways for initiating the death pathways. So it is going to be death receptor pathway or the mitochondrial pathway. In the death receptor pathway, the death receptors are always been present onto the plasma membrane, and they are actually going to receive the signal from the death ligands, and that's how it is actually going to initiate the activation of a series of caspases, and that's how it is actually going to induce the cell death. Whereas in the intrinsic pathway, the cellular factors are going to be released from the mitochondria under the influence of the several the apoptotic inducers, and then these mitochondrial factors are going to activate the different types of caspases, and then ultimately the caspase 3 is going to be activated, and this caspase 3 is going to kill or destroy the proteins, and as well as it is going to activate the DNases, and these DNases are actually going to chew the DNA or the genomic DNA. How you are going to detect the apoptosis? So we have discussed about the morphological parameters, we have discussed about the DNA fragmentations, and we also discussed about how you can be able to flow cytometry to detect the apoptosis into the mammalian cells. So with this brief discussion about the apoptosis and the autophagy, I would like to conclude my lecture here. Thank you.